Here we'll learn how the body metabolizes chylomicrons. To begin, start a table to learn the three key pathways in lipoprotein metabolism. The exogenous pathway, in which chylomicrons clear dietary lipids. The endogenous pathway, in which VLDL and LDL transport and distribute endogenously synthesized lipids, those synthesized in the body. Reverse cholesterol transport, in which HDL clears excess cholesterol. In this tutorial, we'll learn the exogenous pathway. Now let's denote the two key enzymes involved in lipoprotein metabolism, which both degrade triacylglycerol to glycerol and free fatty acids. Lipoprotein lipase, which is bound to the endothelial layer of the peripheral capillaries, and hepatic lipase, which is also a phospholipase, meaning it can degrade phospholipids and localizes within hepatic endothelial cells. We'll only see the lipoprotein lipase in this pathway. Next, let's illustrate the exogenous pathway. First, draw a small intestine epithelial cell, which will house the chylomicron. Then draw representative peripheral tissues and a capillary adjacent to them. Then draw the liver. Now let's draw the exogenous pathway. Make a notation that it only occurs in the fed state. Why is this? Because its purpose is to clear dietary lipids. So let's illustrate this now. To understand the exogenous pathway, we need to first understand what chylomicrons are, which clear dietary lipids. Draw a chylomicron within the intestinal cell. It's the largest and least dense of the lipoproteins. Write that chylomicrons are only synthesized in the fed state, are cleared from the body within minutes, are 98% lipids. Primarily, they are dietary triacylglycerols, TAGs. Secondarily, they are cholesterol esters, CE, and free cholesterol, C. Let's note these abbreviations for reference. Next, draw a semicircular shape on the chylomicron surface to represent an apolipoprotein. We'll use this shape to represent apolipoproteins throughout this tutorial. Label it B48 and indicate that ApoB48 is a marker of dietary lipoproteins. Now we're ready for the steps in the exogenous pathway. Step 1. Dietary lipids are packaged into a chylomicron, which is synthesized in the endoplasmic reticulum. Step 2. Show that the chylomicron enters the lymphatic system, which empties into the circulation via the left subclavian vein. Thus, chylomicrons enter circulation here. Step 3. Draw an HDL. Show that it primarily comprises cholesterol esters and secondarily comprises triacylglycerol and free cholesterol. On its surface, draw ApoE, an E in a crescent shape, and ApoC2, a C2 in a crescent shape. Then show that HDL donates these surface apolipoproteins to the chylomicron. HDL functions as an apolipoprotein reservoir. Then draw the endothelial layer of the capillary. Show that lipoprotein lipase, LPL, which is an enzyme in the capillary lumen, is anchored to this layer. For step 4, ApoC2 binds LPL and activates it. For step 5, LPL degrades chylomicron tag into glycerol and free fatty acids. Show that glycerol returns to the liver, where it can be metabolized to DHAP, a substrate of gluconeogenesis. Show that the chylomicron's free fatty acids enter the peripheral tissues, particularly the mammary glands, adipose, and muscle tissues. Free fatty acids are stored in adipocytes or oxidized for energy in muscle cells to fuel contractions. This step also occurs in the endogenous pathway. Lipoprotein lipase allows circulating lipoproteins to degrade and distribute their cargo. Show that the chylomicron is now a chylomicron remnant. It's primarily cholesterol esters, secondarily triacylglycerol and free cholesterol. indicate that it still contains ApoB48 and ApoE on its surface. But for step 6, show that ApoC2 returns to HDL, which prevents further LPL activation. Draw a remnant receptor on the liver. Step 7, ApoE binds it and the chylomicron is endocytosed. Step 8, the remnant releases its cholesterol in the liver and is finally degraded by lysosomes. Recall that chylomicrons are cleared within minutes, thus this entire process from chylomicron synthesis to remnant degradation occurs very rapidly. As a clinical correlation, write that familial hyperchylomicronemia is a rare hereditary disease in which patients cannot clear chylomicrons efficiently. It can be a result of either LPL deficiency or ApoC2 deficiency. As we have seen, ApoC2 activates LPL. Imagine that we study a healthy patient's blood plasma sample after fast. The blood plasma is relatively translucent. It's cleared of chylomicrons. When we collected blood plasma after a meal, however, the chylomicrons produced give it an opaque, milky appearance. In patients with familial hyperchylomicronemia, a blood plasma sample looks cloudy even after a fast. Lipemia retinalis, the cloudy appearance of capillaries in the retina, is a common symptom of this disease. This concludes our diagram.